Xylella fastidiosa, about also ecology of the vectors, like host-plant associations and implications on spread of Xylella, also about dispersal, and also about acoustic communication, like the use of vibrations to manipulate the behavior of the insect vector. So very interesting presentations. There will be some time. The time for presentations will be 15 minutes, so it should be restricted to the time. And then we have some extra time for questions and answers after each speaker. And then at the end, we'll invite the speakers to be here in front so that everybody can make further questions and can have some discussions. Okay, the first presentation in this afternoon is I will be presenting. So, okay, this is the work of my PhD student, Mariana Boss Esteves. She was not able to come, so I want to present for her. This is her picture on the left, upper corner. This work was done in cooperation between two labs in Brazil, one from University of São Paulo, the insect vector lab, and also the lab, citrus biotechnology lab in Instituto Agronômico, which is coordinated by Alessandra, and we had the involvement of several students working in this project. So as you know, Xylella pathosystems are very different from each other. They are complex. You may have different Xylella subspecies. You may have different sequence types. You have different variation in biology, in genetics, even between a sequence type among different strains. You have different crops affected, many host plant associations, and that has implications on vector species that will be involved. So you have different vector species because of different plant associations and different regions. And so a big question we have talking to people here is how transferable is the information from one system to another, like from one subspecies to another, from one crop or variety to another, from one region to another. Here, I will show that at least part of this information can be transferable. For example, there is a reasonable amount of information on vector bacterial interactions developed especially for fastidiosa subspecies. Studies mostly done in California. And there are studies also for the other subspecies, but they are more on vector competency, on host plant associations, not much on transmission mechanisms or molecular basis for that. So we can have, we can use a lot of the information generated for fastidiosa subspecies and vectors from grapes, and part of it we can use for other systems. Transmission involves three important steps: acquisition, and then when the insect feeds on the infected plant, then the bacteria is retained in the foregut and multiply, forms a biofilm, and then the bacteria can be inoculated by the insect while feeding on a health plant. So here I'm going to focus on the retention, how bacteria is attached to the foregut. So this is apparently simple, but as you know, fluid velocity are very, very high here. It's a very narrow duct, the pre-Siberian. So we have very high flow speeds, and the bacteria needs to have important special mechanisms to attach to the surface in the vector. Adhesins are membrane proteins that are thought to be involved in this attachment, in the early attachment, and also in the biofilm formation. So some of these proteins, they are important to attach to surface polysaccharides, for example, in the cuticle of the foregut. and then form a biofilm by promoting also cell-to-cell adhesion. This is a kind of a conceptual model for colonization of Xylella fastidiosa in vectors. It was developed by Chiellini and Almeida. And initially the first cells are attached, and then you have microcolonies forming. Usually they are laterally attached, and then at some point they become polarly attached, and then at a later stage the biofilm will produce like daughter cells which will form other biofilms. So it's kind of a stepwise process, and different proteins may be involved in several stages of this process. For example, there are two major types of proteins in the membrane. One is type is afimbrial proteins, like pili. Other are afimbrial proteins, like afimbrial adhesins. And afimbrial adhesins are particularly involved in initial attachment, for example. This is a study. I'm going to just make a review of background information on that. So some of these proteins have been tested. For example, hemagglutinin-like genes that code for these afimbrial proteins, like this XAGE, XFA, XFB, were tested for transmission. They produced mutants for bacterial mutants for these genes. They inoculated grapes, and then they measured transmission, saw that the transmission was reduced, so it has an effect on transmission. And they measured the population of the bacteria inside the vectors, and noticed that there was a decrease in the first hours after acquisition, suggesting that affects the initial attachment, so the beginning of the biofilm formation. Other studies used this technique called in vitro acquisition system, which allows the vectors to acquire cultured cells in an artificial diet. This is apparently a very simple process, but it took a long time for people to find out how to get this working. So, for example, the work of Chiellini and Almeida about 10 years ago showed that it was necessary to grow in a minimal medium, and also the transmission efficiency was enhanced by using like a host structural carbohydrate that could trigger genes involved in stickiness of the bacterium. So by growing this bacteria in these special conditions, they can be scraped and placed in an artificial diet, liquid diet. This is offered to the vector in this like a small cage in a membrane sachet, so the vectors can puncture this membrane parafilm sachet and feed from this diet containing the cultured cells, and then they can transmit to plants. This is very important for this type of functional work, because many of these mutants, they cannot grow, they cannot infect plants, or they cannot move systemically, and this will affect transmission. So by using this type of systems, you can work these problems. So another work that used this type of system, they tested the effect of some membrane proteins, and by adding antibodies to the sachet together with the cultured cells, and then these antibodies could be blocking these proteins, and then you could test. And in this particular study, for example, by Chiellini and others, they found the effect of this ZADI, A1 and A2, and hemagglutinin-like genes, which are afimbrial proteins in the blocking transmission rates. In this other study, for example, they transformed the bacterial strains for mutating the genes for these proteins, and they offered these bacterial strains in the diet. And I measured the transmission at several moments after acquisition to evaluate the effect on the initial adhesion, like 12 hours after acquisition, or later on they could have an idea about the effects on retention. So you can see here like genes like RPF that regulate pathogenicity factors, and also some membrane protein genes could affect either both phases, beginning and later attachment, or the beginning of the attachment. So this was for fastidiosa, so a lot of information has been generated. There are other papers too, but for other subspecies, this has not been studied. We have, for example, a very good amount of information for expression of proteins in planta or in vitro for citrus strains of Xylella fastidiosa. Some of them, like FIN-A, ZAD-A2, they are expressed at different stages of biofilm formation in plant and in vitro, for example. But we did not have any information about the role of these particular proteins in the vectors. So in this work, we were testing the particular effect of ZAD-A2 as a blocking agent for transmission by the vector. But to do this, my student had to work on the validation of this artificial diet for the transmission in vitro. And so basically, what we did is to use the same system, try the same conditions, but for the citrus strains of subspecies pauca. So the bacteria is grown in culture for like 10 to 12 days, and then the cells are scraped, and this is in the minimal medium. Here we did not have to add like galacturonic acid or pectin. The efficiency was not shown to be improved for subspecies pauca as it was for fastidiosa. So we only grow on XFM, it's a minimal medium. Put the bacterial cells in suspension in this artificial diet made of amino acids, as published by Quirine Almeida. We tested different vector species, and we noticed that Macugonaria leucomelas adapted very well to the cage and was good at acquiring and inoculating plants. We used like the 6-hour acquisition access period. Here, after this acquisition in the diet, we put the insects for a gut washing, because you need to make sure that the cells that are there in the insect, if you do a PCR, are not cells that are just loose in the gut. They are really attached. So you have to put these insects to feed on a plant, ideally a non-host plant, so that they can lose this bacteria that is not retained. And then, afterwards, you put on a test plant to measure the transmission. So you need good indicator plants. In the case of citrus, we use the Catharanthus roseus. We also had to develop, and this was done by the group of Alessandra Souza, to develop an antibody against the ZAD-A2 protein. This antibody was synthesized for a 34 kilodalton fragment in a previous paper. And this was the assay. So basically 3 treatments, very simple. In the first one, we used the antibody against this ZAD-A2, an afimbrial protein. So the cells were, as shown before in the system, they were grown in vitro, and then they were added to the liquid media in the presence of antibodies against this membrane protein, anti-ZAD-A2, with this dilution. And then the insects were fasted for 1 hour, and then placed on the sachet to feed on this diet, and then gut wash, and then tested in plants. The same procedure was done for the treatment 2, except that here we didn't add the antibodies, just the Xylella fastidiosa and the liquid diet. So this is a kind of a positive control. And we had third treatment was a negative control, where no bacteria was added, just the liquid diet, to make sure that the insects were free also from the bacteria. And this was tested at the acquisition by vectors using PCR, qPCR, and transmission to test plants, putting the insects on the plants, and then 9 days later doing PCR from the plants. And here are the effects on acquisition rate. So this is proportion of insects that were turned out to be positive by PCR for Xylella fastidiosa. So when you have only Xylella fastidiosa in diet, you get like 56% on average of acquisition of positive insects. But if you add the antiserum, you get a 3-time reduction in this rate, up to 18% on average. So there is an effect on acquisition rate, and none of the negative control insects tested positive, which was important. Concerning transmission, here are the percent of individuals that transmitted Xylella fastidiosa to the Cataranthus plants, to our test plants. And you see that the transmission rate by single insects was 10%. And when you add the antibodies to the diet, you get 1/3 of that. So there is a reduction, both in acquisition and in the transmission rate. So in conclusion, we could demonstrate here in this very simple assay that this protein, ZAD-A2, is important for acquisition and transmission. We didn't have a total blocking effect, and this is can be explained by the fact that the other proteins are also involved. So it's a complex process. So you need to act maybe in more than one protein to get a complete blockage. And maybe this ZAD-A2 can be used to block Xylella fastidiosa in a control strategy. It may also be applied to other subspecies. So it was already shown to work for fastidiosa, for pauca, and may be applicable to other subspecies because this is a kind of a conserved protein. Okay, this is what I had to present. Just to acknowledge the funding agents, especially São Paulo Research Foundation, FAPESP, our state agency, and also federal agencies that provide the fellowships to the students. Okay.